Hi, PBio. Last time we saw each other, we started the protein synthesis notes, and each class is about 5 to 6 slides in, so we are going to start on slide number 5, and we'll proceed to the end of protein synthesis, so slide number 17. So we'll start with tRNA. tRNA stands for transfer RNA. It gets its name from the fact that it transfers an amino acid to make a protein chain, a polypeptide chain. So the amino acid will be on one end. So we see right here in the illustration, it has a binding site or attachment site. And then on the other side of the tRNA, we're going to find what is called an anticodon. An anticodon, its purpose is solely to be able to carry the whole entire tRNA to the ribosome and be able to drop off the amino acid. Okay, so the tRNA has an anticodon, which are bases, just like in RNA. So we're going to see A and U and G and C. And the anticodon is the complementary bases to the mRNA. So for example, if the mRNA has AUG, so on the mRNA strand, it's AUG, the anticodon will have A that pairs with U, so this would be U. Instead of the next letter on the mRNA being U, this would be A. And then the last one, G, this one would be C. Okay, so when we think about transcription translation, DNA will always get transcribed into mRNA. That mRNA codon never becomes tRNA. The anticodon is simply an attraction mechanism to draw the tRNA into the ribosome and drop off the amino acid, which in this case, if the mRNA is AUG, the amino acid would be methionine. Okay. The other really important thing we have to focus on when we think about this slide is that the last nucleotide, or the last base here on the tRNA, is flexible, meaning that as long as the first two are the same, the third one will be flexible to attaching an amino acid. So this is where we sometimes lose a couple people, so I'll go back to our previous slide here. And what we notice is there's a chart here with redundancy, a redundancy code. So if we look here at leucine, we're going to find that no matter what the last base is, it's still going to make leucine. As long as CU are the first two bases, the last base is flexible. So that's where the wobble comes in, or the flexibility here on the third base of the tRNA. This will save a lot of resources in a cell if we think about we are going to have the ability to make 20 amino acids. Okay, and so for those 20 amino acids, we're going to actually have 61 codons, or 3-letter triplets, in the mRNA. So if we're able to save and not make 61 tRNAs, that's going to be a lot of resources that don't have to be reused or be inefficiently used in the cell. So we can actually, because we have the wobble, only produce 45 tRNAs. Now these tRNAs are floating throughout the cell and they're waiting for their turn to be used in making a protein chain. Okay, so let's take a look at the next slide. We're going to see here we have the tRNA. It has the anticodon at one end, and it will have a binding site, an attachment site for an amino acid. So what's going to happen is this biochemical reaction. Just like we learned enzymes in our biochemistry unit, they're going to come into play in this unit as well. So we have a set of enzymes. So this gray blob here represents a set of enzymes called amino-acyl tRNA synthetases. So what they do is they use ATP to physically bind this amino acid to the tRNA. So it's going to be a chemical reaction of physically bonding the amino acid to its corresponding tRNA when it's needed. And so each enzyme is going to be unique for the tRNA. So if there are 45 tRNAs, we're going to have this set of 45 enzymes that will do the reaction where they're using ATP to physically bind the amino acid to the tRNA. Okay, so now we're moving on to ribosomal rRNA is the shortened name, and rRNA only has one purpose, and that purpose is to make the ribosomes. If we look closely at the structure of ribosomes, we're going to see a small subunit and a large subunit, and there's an overlapping region, so they kind of bind on top of one another. And in that common space where they overlap, that's where the mRNA will be read. Okay, so the mRNA goes in, we notice in this picture we have 5' to 3', and it's going to get read in this direction. So what's going to happen is the mRNA keeps moving along in the ribosome until we have the start codon that's located, so always AUG. So in the moment where AUG is found on the mRNA, in that moment, we're going to have the first tRNA come in. It's going to come in at the middle site, the P site. This is the only tRNA that ever enters at the P site. All the other tRNAs will enter at the A site. Okay, so there's 3 sites here in order, A, P, and E. So our very, very first amino acid, or methionine, the tRNA will enter at the P site. Okay, and what we'll see is here at the top, it will start forming a polypeptide chain or make a protein. And each protein always has to start with methionine. So let's review the 3 sites here that we briefly skimmed over. So aside from the very first amino acid methionine, the rest of the tRNAs will enter at the A site. This stands for amino acyl binding site. So you see the full name here, amino acyl binding site. That has to do with the actual enzymes that we just went over. After the amino acid gets dropped off at the A site, what will happen is if you think of a conveyor belt, so they're all going to move over one spot to make room for the next tRNA to come in at the A site. Okay, so let's say we've had this tRNA come in at the A site. It will then, the entire tRNA will shuffle over one spot and it will go to the P site. The P site stands for peptidyl binding site. The way that you can remember this or think about this is that at the P site, a peptide bond will form. So the name peptidyl binding site comes from the fact that we have an actual peptide bond formed between these 2 amino acids that are present at the A and the P. Okay, so the amino acids will form a peptide bond just like we learned about in biochemistry. When we think about proteins, the bond specific to polymerizing proteins or binding amino acids together is a peptide bond. If we do a little bit of review now, so when we think about carbohydrates, we think glycosidic linkages. When we think about nucleic acids, we have phosphodiester bonds. So just a little refresher on what we learned about. Okay, so P stands for the peptidyl site. We're going to make a peptide bond between 2 amino acids. Now that the amino acid has fully been dropped off, okay, and the chain is built with peptide bonds, the tRNA that was first in the A spot and then in the P spot is going to move to the E binding site. E simply stands for exit. This is the last binding spot within the ribosome before the tRNA exits and is then reused within the cell whenever that cell needs to bring in the same amino acid back to the ribosome. Okay, so transcription overall has 3 steps, and please do not mix these up with translation. So the names of the steps are actually the same. Initiation is just a general term for start, okay? Elongation is a general term for meaning we're going to build that strand. We're going to lengthen the mRNA or lengthen the protein strand. And then termination is just a general term for stop. So when we think about transcription, you're going to see initiation, elongation, termination. When we then move on to translation, you're going to see initiation of translation, elongation of translation, termination of translation. So on a test or in an assessment, please make sure if you use the term initiation, which process are you talking about? Transcription or translation? So first, when we talk about translation, we are thinking about DNA being transcribed or the bases being turned into mRNA, the message being taken by messenger RNA so it can leave the nucleus and find a ribosome. So 3 steps that happen. First, to start the whole process, what's going to happen is a special enzyme right here called RNA polymerase is going to find a promoter site. This is like a starting site. So transcription cannot happen unless the TATA box is found. So this is simply TATA, so thymine, adenine, thymine, adenine. So this is the starter sequence or the promoter site for transcription to begin. In that moment, when the protein enzyme, RNA polymerase, finds the TATA box, then the DNA will get unzipped by the same enzyme. So it's important to kind of pause along the way and think about what is getting unzipped. So when we think about DNA, what types of bonds are holding the two strands together? So now we're talking about hydrogen bonds. We said phosphodiester bonds are to hold the nucleotides together in the backbone, but between the two strands, we have hydrogen bonds. So RNA polymerase is going to hydrolyze those. It's going to break the hydrogen bonds. Elongation is when the strand is being read, so the DNA strand is being read, and simultaneously RNA polymerase is going to assemble or make the mRNA strand. So in your protein activity, what you did is you looked at the DNA strand and you wrote the basis for mRNA. In real life, what's going to happen is RNA polymerase reads the DNA strand and then builds the mRNA bases that correspond, and it's going to continue building in a 5 to 3' direction. How does this whole process stop? The RNA polymerase keeps building and building and building, and then it's going to find a poly-A sequence. Poly means many, A refers to adenine. So what's going to happen is RNA polymerase is going to reach a segment that has tons of A's. Could be 200 A's in a row, A, all the way through. So that initiates, or that will indicate to RNA polymerase that the termination, the stop sequence is reached, transcription is done, and then what's going to happen is all the factors, so all the enzymes associated with the process are going to be released. Where do they go? They just go to float in the cell until they're needed again. So let's look at this process in a picture rather than words. That's easier to understand. So here at the top, we have the DNA strand. It's going to be a double helix. So we have one strand that is 5' to 3', and the strand below it is also going 5' to 3' in the opposite direction. So this is what anti-parallel means. So we have the start point. Remember, the start point or the promoter is the TA-TA. So this RNA polymerase, this blob here, is going to identify the TA-TA sequence, the TATA box. As soon as it has identified the start or promoter region, RNA polymerase is going to unzip the DNA. So it's going to break the hydrogen bonds. At the same time, it will start to read the DNA, and it will make the transcript. So here, the tiny little pink is the mRNA transcript that it will make based on the complementary bases found in the DNA. Okay, so please remember it's going to build in a 5' to 3'. So in this picture, it has to read the bottom strand, which is DNA that is anti-parallel. And in any picture, if you were to get this on a multiple choice section, a diagram, an open response, don't just memorize that it's the bottom strand that is being read. Look at the mRNA transcript, look at the numbers, and make sure you understand that the strand will get built 5' to 3', adding to the 3' end. And so then you just have to find the corresponding opposite anti-parallel strand on the DNA. So we have 3' to 5' here. It's getting read, and it's building 5' to 3'. Okay, so at the end here, how does this stop? Elongation happens for a very long time until we reach a termination sequence. That is solely a poly-A tail. So many A's all in a row that will indicate to RNA polymerase, stop. We're done with transcription, and RNA polymerase gets released into the cell, and the mRNA transcript gets released. Next, we have mRNA processing. So there are 3 steps before the mRNA can then find a ribosome and start translation. mRNA processing involves the addition of a 5' cap, then a poly-A tail, and then RNA splicing. So in the next video, we're going to start here on mRNA processing and complete the rest of the PowerPoint. Thanks so much for watching.